Hi everyone, welcome back. Today I'm going to take up the last lecture of your semester, and this lecture is the second part on the topic roles of calcium and hormones in bone metabolism. And this will be a relatively very short lecture. We all know that calcium plays many important roles in the uh, cellular metabolism and different aspects of physiology. We may mention the role of calcium as a messenger and its role in the transmission of nerve impulse from one neuron to another, where it is responsible for the fusion of synaptic vesicles to the synaptic membrane, releasing the neurotransmitter into the synaptic cleft. In this particular topic, we are going to see the role of calcium in bone metabolism. Bone, as we have seen, consists of three major components: minerals, organic matrix, and water. And to some extent, some lipids are also there, but very little contribution from lipids. Nevertheless, it has some amount of lipids also. So majority of the bone is formed of these minerals. And what are these minerals? And this mineral, as we call hydroxyapatite, consists mainly of calcium and phosphate. So these two ions makes up the majority of the bone material. So if a bone formation has to undergo, then these two ions has to be there. So bone can be formed from these different components, and bone can be broken down or resolved, giving these components back. So in today's lecture, we will talk on the effect of calcium on these two processes and two hormones, parathyroid hormone and vitamin D. And we will also see how phosphate also has an impact on the two processes of bone metabolism. Now, bone metabolism is nothing but bone remodeling, and bone remodeling is this formation and resorption of bone. First of all, let's see the types, the forms of calcium which are present in our system. Overall, a healthy adult contains around 1 to 1.3 kg of calcium, and of these, 99% of the calcium are in the bone, whereas only 1% are in the soft tissues and extracellular fluid. But we will see later that these 1% of calcium is very important, and depending upon the concentration of calcium, in particular in extracellular fluid, the calcium in the bone will be metabolized in order to maintain the concentration in the extracellular fluid. So coming to this 1% of calcium, and particularly in plasma, the calcium is not present in one single form. It is present in three different forms. Either it may be present as ionized calcium, or it may be bound to protein, or it may be complex with other ions also, or other molecules. As here, calcium is bound or complex to various ions in plasma, including bicarbonates, citrate, and sulfate. Whereas non-phosphate is complex to various cations, including sodium and potassium ions. So this is the normal concentration range of calcium present in the plasma. Is 8.5 to 10.2 milligram per deciliter. That is 100 ml of the plasma contains 8.5 to 10.2 milligram of calcium per 100 ml. And these calcium is distributed in these three forms. So almost 50% of the calcium is in the ionized form, and another 50% is distributed among protein and other anions. Majority is bound to the proteins. So, but the overall concentration of plasma calcium will decide or will have impact on many other physiological process. Let's continue. Now, the two hormones I have mentioned, there is parathyroid hormone and vitamin D, or which we call calcitriol also, or 1,25 dihydroxy vitamin D, regulate plasma concentration of calcium and inorganic phosphate. So the role of these hormones in bone metabolism comes from its role in maintaining the plasma concentration of calcium in particular. Let's see how they are interconnected. So in this particular diagram, we'll see the effect of low calcium, plasma calcium, on the different uh, organs and hormones. So if the plasma concentration of calcium is lower than the normal, then these will be sensed by the parathyroid glands, and as a result it will produce parathyroid hormone. Now this parathyroid hormone has receptors, particularly in bone cells and kidney. In case of bone, it leads to the increase of bone resorption, resulting in the release of calcium and phosphorus into the plasma, and because of this the plasma calcium concentration increases. So when the concentration is increased to a certain level, then it will inhibit the release of more parathyroid hormone. So it is a negative feedback uh, mechanism where the calcium ion concentration again leads to the inhibition of release of parathyroid hormone. The same case is in case of this kidney. In case of kidney, it has two biochemical effects. First, it increases the activity of 1-alpha hydroxylase. The function of this 1-alpha hydroxylase is that it is responsible for converting 25 hydroxy vitamin D into 125 dihydroxy vitamin D, which we call the calcitriol. And this is the hormone which is mainly involved in the regulation of calcium and phosphorus absorption from the alimentary tract. So when this hormone binds to the intestinal cells, then there is absorption of more calcium and phosphorus from your food. So, and these are released into the plasma, and because of that there is increase in the concentration of blood plasma. And if the concentration is increased to a certain level, then it will inhibit the release of more parathyroid hormone. If there is no parathyroid hormone, then there will be no active alpha hydroxylase, 1-alpha hydroxylase, and there will be no calcitriol, and the absorption of calcium and phosphorus will not be efficient. So thereby reducing the calcium concentration in the plasma. So in these two ways, it regulates the calcium. Another effect of parathyroid hormone on the kidney is that it reabsorbs most of the calcium during urine formation. Because of this hormone, there is increased calcium reabsorption, and because of this, the calcium concentration in the plasma increases. But here, the effect of parathyroid hormone on the kidney in case of phosphate is different. Whereas it increases the calcium absorption, it increases phosphate excretion. As you see here, because of this, phosphate excretion is increased. There may be a rationale for these. As you see here, when there will be parathyroid hormone, then there will be increased rate of bone resorption, and lots of calcium and phosphorus will be released. Where calcium is very important and used for many other purposes, the excess phosphorus is of no use, so it increases its excretion. This is one of the reason, but there may be other also. One other feedback mechanism is the inhibition of release of parathyroid hormone from the parathyroid glands by vitamin D. As you see here, the 1-alpha hydroxylase, which was activated or increased activity of hydroxylase, which leads to the formation of more vitamin D. If there is more vitamin D, there will be increased absorption of calcium and phosphorus from the alimentary tract. So it will lead to the increased calcium ion in the plasma. So if it has increased to a limit, then the level more than the normal level, then there is no need to absorb more calcium and phosphorus. So because of that, this hormone again comes and binds to parathyroid hormone and stops production of more parathyroid hormone, thereby stopping these processes. Okay, one question comes up in our mind: how the calcium ion in the plasma is responsible for the production of or inhibition of parathyroid hormone secretion or production. There is a receptor called calcium sensing receptor, which is present in the membrane of parathyroid cells. The function of these calcium sensing receptor, as the name indicates, is to sense the concentration of calcium in the plasma. So when there is a high concentration of calcium, then this calcium binds to this receptor. It is active as a dimer, as you see here. Now when the calcium binds to this receptor, then it activates the associated or coupled G proteins. So we have discussed in details regarding the G proteins in earlier lecture. So particularly GQ and GI are associated with these or coupled to calcium sensing receptor. Once these G proteins are activated, then they will activate a downstream signaling pathway. The output of these downstream signaling is that it inhibits the expression of or transcription of parathyroid hormone gene. So there will be lesser production of parathyroid hormone. Another consequence or outcome of this downstream signaling is that it will inhibit the release of parathyroid hormone into the extracellular fluid, so that there is reduction in the production of parathyroid hormone. But when there is not enough calcium, or it is lower than the normal calcium concentration in the blood, then this receptor will be free and there will be no activation of these G proteins, and as a result there will be no inhibition of the transcription of parathyroid hormone gene. In that case, parathyroid hormone gene will be produced in normal way. I won't go into details of the synthesis of parathyroid hormone. This is a normal process of transcription and translation. But as it is seen in many of the hormones and enzymes, the parathyroid hormone is produced as a inactive precursor as pre-pro-parathyroid hormone. Then it is again converted to pro-parathyroid hormone and finally to the functional parathyroid hormone. Another effect on the parathyroid hormone gene is that of vitamin D. Now you see here, vitamin D being a steroid-based hormone, it can easily pass through the membrane, so it doesn't require a cell membrane bound receptor for its action. So it can directly come and bind to the vitamin D response element on the parathyroid hormone gene. So presence of vitamin D also inhibits the transcription of parathyroid hormone gene, as we have seen in the previous slide. As you see here, vitamin D inhibits the production of parathyroid hormone. So the inhibition is through the inhibition of this transcription of parathyroid hormone. Whereas this vitamin D has a positive effect on the calcium sensing receptor gene, protein gene. So if there is more vitamin D, then there will be production of more calcium sensing receptor protein, because the transcription of this gene is upregulated when there is vitamin D. So in that way there will be more calcium sensing receptor on the membrane. So what is the relation between these in the overall picture of the calcium concentration regulation? Production of more calcium sensing receptor will allow more sensitivity to the change in the calcium ion in the plasma. Here you see here the effect of this serum parathyroid hormone level on the free calcium in blood. As the concentration or amount of parathyroid hormone decreases, or you see like this, when the calcium concentration is high, low in the blood, then there is high serum parathyroid hormone. But as this parathyroid hormone results in the production of more calcium from bone, the concentration of calcium in the blood increases. When the calcium concentration increases, the serum parathyroid hormone level decreases. So it remains at a basal level at high calcium ion concentration in the blood. This is the this different diagram, but it explains the above regulatory mechanisms in a different diagram. So almost the same thing. When there is low calcium ion concentration in the blood, then the calcium sensing receptor detect that there is low calcium. That means there is not enough calcium ion to bind to calcium sensing receptor, and as such there will be no activation of the G protein coupled to calcium sensing receptors, and because of that there will be down regulation of the transcription of up regulation of the transcription of parathyroid hormone, and it will also lead to the excretion of more parathyroid hormone. Then this parathyroid hormone will bind to its receptor present in the bone and in the kidney. In the bone, it will increase the resorption rate. Because of resorption, the calcium deposited in the bone will be released in the plasma, because of this the calcium concentration will increase. Similarly, by a different mechanism in case of kidney, it will increase the reabsorption of calcium during filtration, glomerular filtration, and as a result it will increase the calcium ion concentration in the plasma. In case of kidney, another mechanism is it leads to the production of more 1-alpha hydroxylase, which will convert 25 hydroxy vitamin D into 125 dihydroxy vitamin D, and these vitamin D, which is also called calcitriol, will bind to its receptor in the intestine and will lead to the absorption of more calcium and phosphorus from the food, which will be brought to the plasma. So overall, these different mechanisms increase the blood calcium level. When a calcium level has increased higher than the normal level range, then calcium will have a negative effect on the calcium sensing receptor, so on the production of parathyroid hormone. When there is high calcium ion concentration in the plasma, it will bind to the calcium sensing receptor and the associated or coupled G proteins will be activated, and which will result in the production of inhibition or reduction in the production of parathyroid hormone. So there will be a reduction in the resorption rate, and reabsorption rate also will be reduced. There will be no production of these enzyme and no more vitamin D. This is in case of this nephron of kidney, how the different areas, portions of the nephron are involved in the calcium reabsorption. Again, this is a very simplified diagram which explains or which connects the role of parathyroid hormone in modulating the bone remodeling. So the actual mechanism of action of parathyroid hormone on the bone cell is that it activates or it allows osteoblasts to produce more osteocytes, osteoclasts. Even though so because of parathyroid hormone, there will be production of more active osteoclasts. Now, production of these active osteoclasts is because of the action of parathyroid hormone on the osteoblast cells. Osteoblast will produce some active molecules which will then bind to the precursor or inactive osteoblast, whereby this inactive osteoblast will be converted to the active osteoblast. So in the simplified term, parathyroid hormone will allow osteoblasts to produce more active osteoclasts. So we'll see more in more clear picture in one of the slides. This next slide, as you see here. Now, the formation of this active osteoclast is controlled by osteoblast, and the activation of this pro-osteoclast to this active osteoclast by osteoblast is because of the action of parathyroid hormone on the osteoblast. On binding of the parathyroid hormone on the osteoblast, it will produce this monocyte colony stimulating factor, which will bind on its receptor on the precursor of osteoclast, which is also known as monocyte macrophage lineage pre-osteoclast, and it gets differentiated into the pre-osteoclast. Now, this rank L, which is produced by osteoblast, will bind on the receptor present on the pre-osteoclast, and binding of these to the pre-osteoclast will lead to the activation, which is mainly the fusion of many cells or pre-osteoblast to form a polykaryonic osteoblast, means an osteoclast having many nuclei. So in this way, the activity of osteoclast is controlled by osteoblast. Now, what happens is that in the absence of parathyroid hormone, osteoblast produces another active molecule, a hormone, osteoproteaderine, which will inhibit rank LL, this rank L form binding to rank, and also thereby inhibiting the activation of pre-osteoclasts from maturing into the active form. And you see as there are lots of active because of this activity of this osteoclast, the calcium and phosphate which were part of this bone is released into the blood. That's why there is an increase in the plasma calcium concentration because of the bone resorption. So this is what I would like to share with you. If you have any issues or confusions, let me clear you.